Infant and childhood food allergy, whose cause has long been a mystery, has now been linked to a mix of environmental and genetic factors that must coexist to trigger the allergy, reports a new Northwestern Medicine study. The factors contributing to food allergy include the genetics that alter skin absorbency, use of infant cleansing wipes that leave soap on the skin exposure to allergens in dust, and skin exposure to food from those providing infant care. Food allergy is triggered when these factors occur together. This is a recipe for developing food allergy, said lead study author Joan Cook-Mills, a professor of allergy-immunology at Northwestern University Feinberg School of Medicine. It's a major advance in our understanding of how food allergy starts early in life. The paper will be published in the Journal of Allergy and Clinical Immunology on April 6. Food allergies are on the rise and affect an estimated 4 to 6% of children in the U.S., according to the Centers for Disease Control and Prevention. The prevalence of reported food allergy increased 18% among children under age 18 years from 1997 to 2007. Recent data also show hospitalizations with diagnoses related to food allergies have increased among children. Food allergy risk factors can be modified at home. The good news is factors leading to food allergy can be modified in the home environment, Cook-Mills said. Reduce baby's skin exposure to the food allergens by washing your hands before handling the baby, Cook-Mills said. Limit use of infant wipes that leave soap on the skin. Rinse soap off with water like we used to do years ago. Scientist discovers the perfect storm triggering food allergy. Cook-Mills made the discovery by using clinical evidence about food allergy in humans, the effects of food allergen and environmental allergen exposures, and neonatal mice with genetic mutations that occur in humans. Clinical evidence shows up to 35% of children with food allergies have atopic dermatitis, and much of that is explained by at least 3 different gene mutations that reduce the skin barrier. Cook-Mills used a neonatal mouse model with skin barrier mutations and tried exposing its skin to food allergens like peanuts. The peanuts alone had no effect. Then I thought about what our babies exposed to, Cook-Mills recalled. They are exposed to environmental allergens in dust in a home. They may not be eating food allergens as a newborn, but they are getting them on their skin. Say a sibling with peanut butter on her face kisses the baby. Or a parent is preparing food with peanuts and then handles the baby. Next, she read about skin research studies that delivered compounds through the skin by using soap. I thought, oh my gosh. That's infant wipes. Cook-Mills said. The top skin layer is made of lipids, fats, and the soap in the wipes disrupts that barrier, Cook-Mills explained. Skin problems that occur with skin barrier mutations may not be visible until long after a food allergy has already started. The neonatal mice with the mutations had normal-appearing skin, and the dry, itchy skin of dermatitis did not develop until the mice were a few months old, the equivalent of a young adult in human years. After the neonatal mice received 3 to 4 skin exposures of food and dust allergens for 40 minutes during a 2-week period, they were given egg or peanut by mouth. The mice had allergic reactions at the site of skin exposure, allergic reactions in the intestine, and the severe allergic food reaction of anaphylaxis that is measured by decreased body temperature. A skin barrier dysfunction was necessary for food allergy to develop in the mice, but there is a wide continuum of severe to mild skin dysfunction with eczema or atopic dermatitis, which in its mildest form may simply appear to be dry skin. In patients with skin barrier defects, there are changes in the proteins in the skin that are a result of mutations in the genes. These gene mutations in patients are primarily heterozygous, which means there is a mutation in one of the two copies of a gene. Accordingly, in the preclinical studies, neonatal mice were also heterozygous for skin barrier mutations. The mice were co-exposed to food allergens such as egg and peanut proteins, allergens in dust, house dust mite or Alternaria alternata mold, and sodium lauryl sulfate, a soap present in infant cleansing wipes. These novel animal studies provide a basis to test interventions that will more effectively block the development of food allergy in infants and children, Cook-Mills said. She is currently studying molecular responses in the skin that are unique to this combination of genetics and skin exposures. The goal is to determine unique signals in the skin that occur during development of food allergy. This will lead to approaches to intervene with those skin signals and block the development of food allergy.